Hi, I'm Zach Stellato. I worked with Dr. Ting Lee and Dr. Eldad Afik on image classification of calcium signaling in the Arabidopsis thaliana. Um, so the Arabidopsis thaliana is a flowering plant native to Asia and Europe. It is self-pollinating, which makes it easy to grow in labs. And as you can see at the apex of the shoot is the meristem, where the new buds, known as primordium, emerge. Dr. Lee uses a confocal laser scanning microscope to photograph the calcium signals of the Arabidopsis over time. It's taken at 1 frame per second, but over here on the right, you can see it's sped up a little bit. The light spots are indicated by the plant cells constantly lighting up in what we call twinkles. They are signals caused by an increased concentration of calcium. At certain times, you can see a bunch of cells lit up at the same time, and that's what we call an oscillation. By looking at a single frame, we can better see the part that we're working on and how we can classify these images. The goal of this project is to train a computer to be able to identify the different features present in the Arabidopsis so that the plant's natural calcium signaling can be quantified into data. This will allow Ting and other biologists to find patterns in the seemingly random activity of the plant. So how do you teach a computer? Well, unlike humans, computers can't recognize photos. They can't tell the difference between a B and a 3, even though it seems trivial to us. So to teach a computer how to analyze an image, we need to use something called machine learning, specifically in this case, random forests. Doing this, we create tests for the computer where we tell it, this is a B or this is a 3. The machine learning process runs until one robot can complete the test we give it with the highest possible score. No one truly understands how the computer can do this, but it does. Luckily for us, Elastic, the program that we're using in this project, does all the machine learning for us, and we just need to design the tests. Before we test, however, we need to give the computer images it can learn from. To do this, our goal is to make the features in the image as distinguishable as possible. We used a method of image subtraction through a program named Fiji in order to reduce any challenges the computer might face. For example, in the left side of the image, you can see that parts of the microscope reading this calcium signal throughout the entire process, when there's actually no calcium signal there. In order to reduce this, we did, on the right, as you can see, image subtraction. By doing that, it only shakes the parts that have changed since 20 frames before. So we take an image and then subtract 20 frames before that. And in that way, we can reduce parts that stay consistent throughout and only get the parts that are changing, or the twinkles and the oscillations. So especially in the one on the right, you can see that the black spots are represented by where there was a twinkle or an oscillation. And the white spots are when there now is one, but there wasn't one 20 frames before. So the elastic physical classification works where we import an image or selection of images that we've already done the subtraction method on into Elastic. We can then choose from different feature sets and help Elastic to look at the image through a variety of different lenses. Then we start drawing. As you can see, I labeled different aspects of it. So the initial twinkle is when a twinkle first appears. And that is, I drew it in yellow, but that is white on this image because it was there, it's now there, but it wasn't there 20 frames before. So you can see the image subtraction taking place. The final twinkles are in dark because there now isn't an image or a twinkle where there was a twinkle. And then the cell I labeled in blue, the background in teal, and the oscillation in green. This is, in this way, we are kind of setting up a test for the computer to be trained on, because then the machine learning goes through the process of giving computer this image with these inputs and saying, can you get it correct? And when one computer can get where I put a yellow dot and where I put a red dot correctly, that's extra points to that program. At the beginning of my annotation process, I thought the more I told the computer, the better it would do. I would go one by one, perfectly tracing exactly in the lines of every single image. but it was still not working that well, and it took a lot of time. I felt like an artist, and that's not the point of this. So I designed an experiment. I selected 2 frames. On the first frame, I would annotate by labeling the features in ilastic to varying degrees of precision. Then I would allow each program to make a prediction based on what I annotated in the first frame, about what frame 2 would look like. Much to my chagrin, the least annotated version actually performed the best and caught 6 final twinkles and 2 twinkles, where the medium only caught 6 final twinkles, and the fully annotated prediction only got 3 of them. I think the main reason for this discrepancy is that when I was labeling the entire image, a lot of the time I was coloring over parts that I didn't notice. And in that way, I was teaching the computer incorrect things. For example, you can see down here at this bottom part that there's actually a final twinkle that I missed. And in this example, I would have actually written over it on the maximum. And that would hurt its abilities to process that in the future because the programs would be trained to ignore that. Whereas in this one, when I left it blank, the computer was able to figure this out for itself. Actually, a lot of the times the computer's a lot better than me. Here are 2 versions of the same image. On the left, we can see the subtracted one, where we input this into the program. And then on the right, I can see the prediction the computer made. A lot of the time, it's very difficult to distinguish, but there's a difference between the oscillation and the twinkle. An oscillation where many light up and where a single one lights up. The computer's pretty good at this, but sometimes it still messes up. For example, in this instance, where the first frame is when a twinkle is first coming into view, and in the second frame, there's a full twinkle. But the computer recognized that as an oscillation because it's a dim version of the twinkle, very similar to how an oscillation is. So although the computer was correctly recognizing the faintness of the signal, as that would generally indicate an oscillation, it's still incorrect. So now we face the challenge of not wanting to decrease the computer's ability to correctly predict oscillations. But I also don't want it to miss this and think that this is an oscillation when it's actually a twinkle. Luckily, I can test this. First, I tested it on 1 frame and I manually annotated this. So I adjusted it by drawing over it to correct it. Then I compared the 2 where I took a prediction on a separate frame before I added that annotation and then after. The main difference is right in here, where you can see that in the second time after I annotated that, 2 little yellow dots or initial twinkles pop up. Luckily, this is actually okay because in this case, I plan when I export to the images to remove any twinkles that are smaller than a certain size. And in this case, these would actually disappear. So they wouldn't be recognized as a twinkle because they're just part of the oscillation. Here are some examples of the predictions that the computer made after only a few hours of annotating. As you can see, it correctly picks up on the final twinkles and the initial twinkles, and actually does a lot better job in places where it'd be difficult to see. So in order to test the accuracy of the computer's prediction, I performed a test on a series of random frames that the computer had never seen before. I annotated what I thought was correct, and then I ran a computer prediction on the same frame and compared the results. Using an error matrix allows me to differentiate when I say that there is a twinkle and the computer thinks that there isn't one, or when the computer says that there's a twinkle, but I say that there isn't one. The data shows that the prediction was mostly correct, but it sometimes listed twinkles that I did not include in my manual annotation. It is worth mentioning that often the computer would find things that I didn't actually see. And when I looked back, the computer would actually be correct. Here are the final results after I tested this on 9 individual frames. Although it's definitely not perfect, it is due to the accuracy given the amount of time I put into it, as I, at the last minute, changed to this. It's actually very impressive. And with just a little bit more work, it would get a lot better. Specifically for this data set, I annotated about 5 frames where I picked out every detail, and with a minimal amount of drawing, and then I did about 10 small corrections on different frames. And with that, even just that limited amount of work, I'm very satisfied with the outcome. Going forward, I would do a lot. I mean, unfortunately, my internship is ending today, and I'm going to be hiking. But if I did have more time, I would continue to fine tune the image classification by adding more annotations and correcting false predictions. I will also test the prediction on new sets of images and refine the output. Finally, I would export the results into a data set that could be used and worked on by organizing it into quantitative data to identify patterns mathematically. Thank you.